Let's get going. Sorry, having some technical difficulties. All right, everybody, let's get started. It's having a little bit of technical difficulties, but should be working now. So today we're going to get started with chapter 12, which is all about enzyme kinetics, inhibition, and control. So first we'll get started with reaction kinetics. And I guess before we sort of get started diving into it, I'll just start off by saying this is really our last big math chapter. So we did a bunch of math at the beginning of the semester. I know math is not everyone's favorite, but we'll get through it together. It's just numbers and graphs, so we'll be okay. So with that, let's get started just talking about what kinds of reaction conditions or concentrations can influence the rate of a reaction. Anybody can just throw out just anything they can think of what might influence a reaction rate. Yeah, absolutely. So concentration of substrate, concentration of enzyme, anything else? Yep, temperature, absolutely. Yeah, pH, Anything else? Yeah. Sure. I mean, I guess that kind of counts as an enzyme, but I'll say or other catalyst. Anything else? No, that's sort of just like a property of the, yeah. Any other stuff you might throw into a reaction or that might turn up during a reaction that could affect its rate? Yeah. Well, that's just another word for substrate, but yeah. Good thought, though, yeah. I mean, you can include that under enzymes. I know it's kind of hard for you to read my mind, but any other ideas? Yeah. I feel like that would just, if oxygen's a substrate, then yeah. I mean, that's part of it. Well, no. I mean, spontaneity does not influence the rate of a reaction. It just influences the direction of a reaction, right? Yeah. I don't know. Yeah, I mean, that's true. I guess it sort of goes under catalyst, but, or it's like under the same sort of thought process. But yeah, that's definitely true. Yeah. Yeah, that's a good one. Inhibitors. So if you have a lot of an inhibitor, that can slow down a reaction. Don't have an inhibitor, or you have little of an inhibitor, that will slow it down less. Yeah. Surface area. I mean, I think that sort of goes under like substrate and enzyme availability, more so than necessarily surface area. Yeah. I mean, there's sort of one other major one that I won't try to pull out of you, but that's how much product accumulates, right? So as more product accumulates, it's going to want to start stimulating the reaction to maybe start going the other direction, right? So the more product you have as you use up substrate, the reaction is going to slow down because it's approaching its equilibrium. And we also talked about, or we will talk about, how products themselves can act as inhibitors to the enzyme or other enzymes in a reaction pathway. So yeah, those are the big things that are going to influence the reaction rate. Any questions about these kinds of stuff? All right. So here are two graphs. They look similar, but they are not the same. They are in fact different. So I guess everybody just take a minute and look at these two graphs. Think of how they are related to each other, how they're different from each other. One or both of them might have a special name for the type of plot that it is. So take a couple minutes to think about that, and then we'll talk about it together. All right, so can somebody tell me something similar or different about these two graphs? Yeah. Yeah, you have the right idea. So the plots are plotting different ideas. So they're both looking at a reaction that's catalyzed by an enzyme. The one on the left, on the x-axis, you have time. So we're looking at what's happening over the course of time. And on the y-axis, we have formation of product. And so you can think of this graph as being called or it's not necessarily called this, but you can think of this as plotting the reaction progress. So here we're looking at, in the beginning, there's no product. Over time, more product is formed, and it's comparing how fast that happens with no enzyme and with an enzyme. But ultimately, they both reach the same point. They reach some point of equilibrium where some amount of product is formed, and then they kind of level off. What do we see happening on the right? Do we see the same thing? Are we looking at the progress of a reaction? Yeah. Yeah, exactly. So on the left, we're looking at one particular reaction. On the right, we're looking at a whole bunch of different reactions. Each point on this curve is a different sort of reaction setup with a particular concentration of substrate. And depending on how much substrate there is, the reaction will proceed at a different rate. So and does anyone know the name of this type of plot? Yeah. Yeah, it's an MM plot or a Michaelis-Menten plot. you can feel free to call this an MM plot because Michaelis-Menten is a lot to say. So yeah, so on the MM plot, we're seeing the reaction rate is on the y-axis. So the more substrate we have, the more quickly the reaction proceeds. On our reaction progress plot on the left, how can we see the rate on that plot? It's not on one of the axes, right? So does that plot give us any information about the rate of the reaction? Yeah. Say that again? Yeah, so what about what information on the plot can give you the rate? What kind of math would you want to do? The slope. Yeah, exactly. So the slope of this line can give us the reaction rate. So the initial reaction rate will be the slope at the beginning of the reaction. And you can see that over time, the reaction is slowing down, and you can also see that the slope is getting flatter and approaching zero. So the reaction starts out fast, and over time it gets slower as product is accumulating. On the right, we're just seeing sort of that initial reaction velocity. And this reaction is also approaching, or not reaction, this Michaelis-Menten plot is also approaching some kind of asymptote, right? What does that asymptote that it's approaching represent? What is the biggest possible y value you could see on this graph? Yeah. The maximum velocity, exactly, yeah. Does anybody know anything else informationally helpful that we can get from our MM plot using the information on the plot? Yeah. Yeah, the Km, right? Yeah. Yeah, perfect. So at 1/2 of maximum velocity, which is, I don't know, maybe around here, if you drop down a vertical line from there, we can get the substrate concentration that gives us half maximum velocity, which we also call the Km. And just like when we were talking about myoglobin and hemoglobin, the Km can tell you information about how strongly the substrate likes to bind to the enzyme or how much they want to interact with each other. So for a small a Michaelis-Menten plot that has a low Km means that a relatively small concentration of substrate is needed to get to the half maximum reaction rate, meaning there's a pretty strong substrate and enzyme interaction. If you have a large Km, you'd need a much larger substrate concentration to reach that half Vmax. So that means there's a lower maybe binding affinity for the substrate to the enzyme, just like we saw with myoglobin and hemoglobin, right? Myoglobin had a smaller KD. It had a much stronger binding affinity for oxygen. Same idea here. And what shape is the curve on our Michaelis-Menten plot? Yeah, it's hyperbolic. Yeah. Sometimes we can also see sigmoidal curves. It depends on the enzyme that we're looking at or the reaction that we're looking at. But in this case, nice little hyperbolic curve. Any questions about these types of plots kind of in general before we dive into it? Yeah. I don't know if it's exactly hyperbolic. It might be some other similar shape. I don't want to say for sure. I should have looked that up. It probably is, but it might be like logarithmic or like something else weird. I should have looked that up. Yeah. Yeah. Yep. Yeah, and the reaction progress plot, the shape also probably depends on the exact reaction that's happening too. just like the MM plot. The MM plot can be sigmoidal if there's, cooperativity in the binding. Same idea with hemoglobin versus myoglobin. All right, so let's start talking about some of the information that we can get out of our MM plot. So here's a little table. So there's these 5 different parameters that are all different types of information that you can get from the MM plot. And so I want you to start thinking about what each parameter is telling you about the reaction. And if you have a fast reaction, would that parameter have a high value or a low value? So go ahead and start filling in that table on your own. You can talk to a neighbor too if you like, and we'll come back together and discuss it as a group in a few minutes. Yes. Yeah, if we don't finish this chapter today, we'll finish it Monday. And then Monday if we're not done with 12, we'll finish 12 and start 13. We may or may not get to 14 before break, but yeah, I haven't posted the quiz yet, but I will get it posted as soon as I can. The quiz for 12 is due in like 2 weeks, like after break, the Monday after break. So there's a quiz due this coming Monday on 11. The quiz for 12 is going to be due the Monday after break, and I will try to get that posted, if not tonight, then tomorrow. Yeah, no, you can do it over break for sure. Yeah. The quiz for this chapter. Yes. Yep. She's thinking really hard about KCAT. Yeah. All right, so let's start filling in our table. So can anybody tell me what Vmax is telling us about a reaction? Yeah. Yes, the maximum rate of a reaction. And that's going to happen when the enzyme is completely saturated with substrate. The fastest a reaction can possibly go is when every single enzyme has substrate in it, right? And so, yeah, so for a fast reaction, would you want a high Vmax or a low Vmax? High Vmax, that is correct. What about Km? What does Km tell us? Yeah. Yeah, exactly. And do we want a high Km or a low Km for a fast reaction? Yeah, a low Km, exactly. Just like we just chatted about. A low Km means there's a relatively high affinity for the enzyme to the substrate, so the reaction can proceed more quickly because the substrate and the enzyme are going to want to interact with each other. And so another way to think of Km is kind of like the Kd that we saw for hemoglobin and myoglobin and the other. We talked about that for another enzyme too, and I cannot remember off the top of my head what enzyme that was, but that's fine. So yeah, so what about KCAT? What does KCAT tell us? Yeah. So catalytic efficiency is one of these other ones. KCAT is involved in catalytic efficiency, but KCAT by itself is telling us something a little bit different. Anybody? Yeah, kind of. I mean, it's the KCAT is the maximum rate per one for an enzyme. So, oops, I gave that away. So, if the KCAT is the maximum rate for an enzyme, would we want a high KCAT or a low KCAT in a fast reaction? A higher maximum rate or a lower maximum rate for an enzyme? Would that make? Yeah, higher. There we go. High KCAT means the reaction will go faster. And KCAT is defined as Vmax divided by the total concentration of enzyme. So it's just telling you for any one enzyme what is its maximum rate compared to Vmax by itself, which is the maximum rate of the reaction with all the enzymes in your container. So what about 1 over KCAT? Yeah. Yeah, basically. So the 1 over KCAT is the what we call the turnover time. So that's the time it takes for one enzyme to make one product. So would we want a high turnover rate or turnover time or a low turnover time in a fast reaction? Low, exactly. Thus, the less time it takes, the faster reaction is happening. All right, so that brings us to KCAT over KM. So what is that one telling us? Yeah. Yeah. Yeah, so the catalytic efficiency is accounting for both the maximum rate and the binding affinity, right? Because KCAT is the rate, KM is the binding affinity. And so if in a fast reaction, would the catalytic efficiency be high or low? High, yeah, awesome. So yeah, the catalytic efficiency, like I said, is taking into account the maximum rate as well as the binding affinity. And the catalytic efficiency is a good way to compare different enzymes or different types of reactions to each other because they account for both the rate and the binding affinity. The other parameters are really good for looking at one particular reaction, but if you're trying to compare different ones, catalytic efficiency is a good way to do that. Okay, any questions about these parameters? You're just thinking, why is there so much math now? Well, apologies, we're going to do some math. So here is a little practice plot. And so using this plot, which was collected with an enzyme concentration of 0.020 millimolar, please go ahead and find the Vmax, the Km, the Kcat, 1 over Kcat, and Kcat over Km. And so I'll give you another few minutes to work on this. Again, please feel free to work with a neighbor, and we'll come back together and discuss it in a few minutes. right. So let's start with the first one. So how can we figure out Vmax from our MM plot? Yeah. So that was in our reaction progress plot. So this is our MM plot. Yeah, how do we find the Vmax? Yes. Yeah. So what would you say is the Vmax for this plot then? Yeah, so that's a really good point. So you can imagine that this plot kind of goes on forever, and it's still kind of increasing a little bit. So the Vmax, and also the Vmax is you never get there, right? It's an asymptote. So you always know it's a number larger than the largest y value you see on your plot. And so, yeah, so we don't, and so you can sort of guess it's somewhere around 500. It's actually impossible to tell because the graph never gets there, right? So just looking at a graph, it's really hard to figure out the Vmax. So yeah, a good guesstimate for that is 500 millimolar per second. And you would know that if you had like the equation for this curve, but without having that equation, you can't tell just by looking at it. So, which is why we have other types of plots that we'll see in a few minutes that help us calculate the Vmax more exactly. All right, so what about our Km? Yeah. Okay, so what would it be on this one? Yeah, exactly. It's right about 10 millimolar. So in case anyone isn't sure, if our Vmax is 500, our half Vmax is right about 250 to maybe about here. And then if we drop down our vertical line, we're right at 10 millimolar. All right. How about our Kcat? Yeah. Yes. How did you calculate that? Yeah, exactly. So we have our V max divided by total enzyme concentration. We know our Vmax is 500 millimolar per second. Our concentration of enzyme is given right here, 0.020 millimolar. So we have 25,000. And what units is that? Yeah. Exactly. Yeah, it's 1 over seconds. Another way you can write 1 over seconds is seconds to the minus first power. Either way is correct. Okay. You don't see that color, right? Yeah. All right. Time to find another color. So how about our next parameter? So we have 1 over Kcat. How do we get that one? That one should be fairly straightforward to calculate. It's right there in the formula. Yeah. Yes. I think that's the right number of zeros. So 1 over Kcat is just 1 over 25. Let me write that in a normal way. Hold on. 1 over 25,000 inverse seconds. And that's going to give us 4 times 10 to the minus fifth. And what units would that be? Seconds, yeah. 1 over Kcat is our turnover time. That's in seconds. Kcat is our turnover rate. That's reactions per second, right? And okay, so that brings us to our fifth one, which is going to be another color. All right, K cat over KM. So we already figured out kcat and km, so this should be pretty easy. Just plug them in, right? So kcat is 25,000 inverse seconds. Km is 10 millimolar. So what does that give us? Yes, 2,500 what? Yeah, exactly. It's millimolar minus 1 seconds to the minus 1. It's kind of confusing to think about those units, but that's the catalytic efficiency. So we have 25,000 like reactions per millimolar per second. Any questions about how we calculate any of those and or where the units come from? All right. So, yeah, so that brings us to our next section, which is about enzyme inhibition. So, I should be able to click here. So now we have another graph. This one looks a little different than our Michaelis-Menten plot. So first of all, what kind of plot is this? Anybody remember the name of this one? Yeah, the Lineweaver-Burk plot. Yeah, and that's another long name. You can call it an LB plot. But that's Lineweaver-Burk plot. So this type of plot is related to our MM plot. How is it related? Yeah. Yeah, exactly right. So in our MM plot, our y-axis was reaction rate. On our LB plot, the y-axis is 1 over reaction rate. In our MM plot, the x-axis was substrate concentration. Our LB plot, the x-axis is 1 over substrate concentration. So we call this a double reciprocal plot because both axes are reciprocals of the MM plot. What is nice about this type of plot? It's linear, yeah. So because it's linear, it makes it a lot easier to calculate, especially our Vmax value. So on our MM plot, the Vmax was this asymptote that we never approached. It's hard to see. On the LB plot, the Vmax is pretty straightforward to calculate. So, yeah, so as I mentioned, this is a linear graph, and the equation for the line in an LB plot is this equation. So I know that's a lot of symbols, but if you break it down, it's just a y equals mx plus b equation. So y is our 1 over v naught, that's our y-axis. X is 1 over substrate concentration, that's on the x-axis. The slope is m, which is km over v max. And the y-intercept, the b, is 1 over v max. So now it's a lot handier or easier to figure out, especially our Vmax, right? And also even our Km. So again, I will give you a few minutes now to calculate those same 5 parameters. And we'll come back and discuss them together in a few minutes. Oh, I should also, sorry, there's the equation for the line, my bad. That will help you. So the actual linear equation is y equals 10x plus 0.5. So now you don't have to count stuff on the graph. All right, so let's get started with the Vmax. So what information on this plot can tell you something about Vmax? Yeah. Yeah, exactly. So our y-intercept, our b, is 1 over Vmax. So our Vmax would be 1 over b, right? Just the reciprocal of the y-intercept. So what is our b in this case? Yeah. Yeah, the answer is 2. But yeah, so we have 1 over, and b, our y-intercept is right here, right? It's 0.5 seconds per millimolar. Not my best millimolar, but you got the idea. And like you said, that's 2. And what would our units be? Yeah, exactly. 2 millimolar per second, just like our Vmax on our other plot. Those are the units that we're going to see for Vmax. It's a rate. So, great. So, how can we figure out our Km? What information on the plot can tell us Km? It's not the y-intercept. Yeah, the slope. Yeah. So, right, our slope is the m in our line equation. So, and our slope is defined as Km over Vmax. So, if our slope is Km over Vmax, you can multiply both sides by Vmax to solve for Km, right? So we have slope times Vmax equals Km. So what is our slope of the line? What number? 10, yes. And what units are that? It's kind of tricky. So remember, our slope is always y over x, right? So our units for y are seconds per millimolar. Our units for x are 1 over millimolar. So overall, if you do seconds per millimolar over 1 per millimolar, yeah, it's seconds, exactly. And then what's our Vmax that we just calculated? Yeah, it's 2 millimolar per second. So if you multiply those out, you get 20. Yeah, 20 what? Almost. Seconds cancel out. Millimolar, yeah. So that's our Km, which we get from our slope. And all the rest of them, kcat, 1 over kcat, and kcat per km, those all come from the same way we did them before. We don't need to go through that again, but it's the same idea, right? Once we have our Vmax and our Km, we can figure out these other ones. All right. Any questions about how we got those? Great. So let's, we'll stop doing math for a little while. Let's talk about enzyme inhibition. So there are 4 different statements here. Each of them describe a different type of inhibitor. So take a couple of minutes and match them up. And then we'll talk about them together. Okay, so let's start with competitive. Which statement describes a competitive inhibitor? Yeah, exactly. So a competitive, oops, I can't write today. A competitive inhibitor is one that binds to the enzyme's active site, and it is often structurally similar to the transition state. It's competitive because it's basically competing with the actual substrate at the active site of the enzyme. So which one of these is the opposite of competitive? Yeah. It is actually not non-competitive. It's the other one. Yeah. Uncompetitive. Yeah. And which statement describes an uncompetitive inhibitor? It is not 2. It's 3. Yeah. This is tricky only because the names are really similar. So competitive is pretty straightforward, and then from there it's like, okay, you got to keep track. So the opposite of competitive is uncompetitive. In uncompetitive inhibition, the inhibitor is going to bind to the enzyme substrate complex, and it interferes with the conversion of substrate to product. So like for an uncompetitive inhibitor, there is no issue with the substrate binding in the active site of the enzyme. The uncompetitive inhibitor is sticking itself somewhere else on the enzyme, and it is stopping the reaction from happening. It's stopping product from being made. And that's sort of the opposite of competitive because there's nothing's happening at the active site for an uncompetitive inhibitor. So, so let's move on to mixed inhibitors. Which of these last 2 statements matches up with the mixed inhibition? Yeah, it's 1. Change up my colors here. 2, mixed is B. So a mixed inhibitor binds to both the enzyme and the enzyme substrate complex. So just like it sounds, a mixed inhibitor is a mix of a competitive and an uncompetitive inhibitor. It does a little bit of both. It binds to the enzyme, it blocks the active site. It also binds to the enzyme substrate complex. So that leaves us with non-competitive. And that is, I won't even ask, that one matches with statement 2, right? A non-competitive inhibitor is a special type of mixed inhibitor. So for a non-competitive inhibitor, it binds to both the enzyme and the enzyme substrate complex with equal affinity. So it's a type of mixed inhibition, because in mixed inhibition, there's binding to both the enzyme and the ES complex. But maybe in mixed inhibition, you don't know if that inhibitor binds to one or the other more strongly. It might bind to the enzyme more strongly by itself, or it might bind to the ES complex more strongly. For a non-competitive inhibitor, it's a specific type of mixed inhibition where it likes the enzyme by itself and the enzyme substrate complex exactly the same. So those are the 4 types of inhibitors we're going to talk about. I'm sorry that the names are so confusing. I did not name them. If I did, they would have been called something else. But anyway, that being said, let's move on to our little table. So here's a table that has those 4 types of inhibitors or types of inhibition, and start to think about for each type of inhibition, what does it do for the Vmax, the Km, the y-intercept, and the slope for a reaction. So you're looking for, does it increase or decrease? And just as a reminder, the y-intercept is 1 over the Vmax, that's written right up there, and the slope is Km over Vmax, and that will hopefully help you figure those ones out. So again, I'll give you a few minutes to start filling in the table. Feel free to talk to a neighbor, and we'll go over it together in a few minutes. Alright, so seems like most people are maybe done. So let's start filling this one in. So can anybody tell me about competitive inhibitors? What do they do to the Vmax? Yeah. Yes. So competitive inhibitors have no effect on the Vmax. So remember, a competitive inhibitor is interfering with substrate binding to enzyme. So it makes it harder for the substrate to get in there, but once the substrate is able to interact with the enzyme, the reaction is able to proceed at up to its maximum rate. But what might that tell you about the Km if there's something competing with the substrate at the active site? And Km tells you about binding. Yeah. Yeah, so Km is going to increase. Remember, Km is that readout of binding affinity. The higher the Km, the more substrate you need in order to get to that half maximum reaction rate. So that tells you about Vmax and Km. So now we should be able to figure out what that's going to do to the y-intercept and the slope in our LB plot. So y-intercept, like we have up here, is 1 over Vmax. So if Vmax doesn't change, what does that mean about the y-intercept? It also doesn't change. Yeah. And what about the slope? Yeah, so the slope is going to increase because our Km is increasing. And if slope is Km over Vmax, if you make the numerator refraction bigger, the whole number gets bigger, right? So that is competitive. How about uncompetitive? So uncompetitive inhibition is when our inhibitor interferes with the enzyme substrate complex. So, and it interferes with the formation of product. So what might that do to the Vmax of a reaction? Yeah, the Vmax is going to decrease because if the enzyme is struggling to make more product, the reaction rate is not going to be as high as it could have been without that inhibitor there. But what does that mean about Km, which is just talking about substrate interacting with enzyme? Does uncompetitive inhibitor interfere with that? So it decreases what? Sorry. The Km? Yeah. So it also decreases the Km. So that in terms of binding affinity, that means the opposite, right? A lower Km means a stronger binding. So why might that be? And so that's because there's what's it called, an equilibrium, right, in between substrate forming the ES complex with the enzyme, and then the inhibitor forming an ESI, enzyme substrate inhibitor complex, with the ES complex. So the more, let me draw that somewhere. So we have substrate is in equilibrium with the enzyme substrate complex, but at the same time, the inhibitor is sort of trying to make this ESI complex. Not my best drawing, but so the more inhibitor you have sort of is going to shift this equilibrium this way towards ESI. And then what that's also going to do is pull more substrate into the ES complex at the same time. So overall, that kind of means that it's going to appear like you have a stronger binding affinity because you need overall less substrate concentration to get to that half Vmax rate. And so now that we know our Vmax and our Km are both decreasing, what does that mean for our y-intercept? Our y-intercept is 1 over Vmax. If Vmax is lower, what does that mean for the y-intercept? Yeah, it's bigger, exactly. So if the denominator of a fraction is lower, the whole number gets bigger. And what does that mean for our slope? Yeah. So that one's a little tricky, right? Because Km and Vmax are both decreasing. And they happen to decrease proportionally in this type of inhibition, so the slope actually stays the same. So let's actually do non-competitive next, because non-competitive is more specific. It's a specific type of mixed, and it'll be easier to do mixed once we think about non-competitive. So non-competitive is an exactly equal mix of competitive and uncompetitive, right? So what would that mean for our Vmax? If for our competitive Vmax doesn't change and an uncompetitive Vmax goes down, what would that mean for non-competitive? Yeah, it goes down, yeah. And how about Km? For competitive, it goes up. For uncompetitive, it goes down, yeah. And for non-competitive, it stays the same. Yeah, it's an equal mix. And how about our y-intercept? It also goes up because we have an equal mix of going up and not changing. Overall, it goes up. And how about our slope? Yeah, exactly. It's still an equal mix of going up and not changing. So that means overall it's going to go up. So, that brings us to mixed. So mixed is a little bit more general. So we have a mixture of competitive and uncompetitive, but it might be a little bit more competitive than uncompetitive. It might be a little bit more uncompetitive than competitive. So let's think about what happens when we mix them together. So in the case of Vmax for mixed, we're mixing together still a no change and a decrease. So overall, what would that mean? Still a decrease. What about our Km? We have competitives going up, uncompetitives going down, and we don't know which one is contributing more, right? It depends on the exact reaction, the exact inhibition we're talking about. So in this case for mixed, it depends. Depending on the reaction and the inhibitor, sometimes Km will go up, sometimes Km will go down, sometimes Km won't change. Mixed is more general, like we were saying. So what about our y-intercept? If we have a mix of not changing and going up, what will that give us? So we have in one of these not changing and one of these going up. If you mix them together, yeah, it's going up. And what about our slope? It's also going up. Thank you. Does anybody have any questions about how these work? Yeah. Yeah, so we have a mixture of competitive and uncompetitive. In competitive, our Km is going up. In uncompetitive, our Km is going down. For a mixed inhibitor, it's a combination of competitive and uncompetitive, but it's not necessarily an equal mixture. It might be a little bit more competitive. It might be a little bit more uncompetitive. So depending which one contributes more, it could go up or it could go down, or it could stay the same, in which case it's non-competitive.
Parameters are always covered.
Yeah, so for these 4 types of inhibition, these parameters will always be like this. Any other questions? All right, so let's put that to work. So we have a graph here. So we have, here is a Lineweaver-Burke plot for some reaction. So take a moment and think about the 3 types of inhibitors, except for mixed. So we have competitive, uncompetitive, and non-competitive. And draw lines on this graph that will represent each type of inhibition. And I'll give you a few minutes to think about it, and then we'll talk about it together. I'll check my colors, blue and purple. And we just talked about how the y-intercept and the slope are changed in each of these inhibitors, so you can use that information to think about what happens to this line, right? So who can tell me for the competitive inhibitor what the line would look like in terms of y-intercept and slope? Yeah. Yeah, exactly. So same y-intercept, higher slope. So start at the same y-intercept, slope is a little higher. That's competitive. How about uncompetitive? Yeah. Yeah, exactly. Higher y-intercept, but a same slope. So that would look something like this. Tried my best to keep the slope the same. You got the idea. And how about pure non-competitive inhibition? Yeah. Yeah, exactly. So higher y-intercept, higher slope, something maybe like that. Any questions about how that would work? Sorry, these colors are not. I tried. Yeah, this one, okay. Non-competitive. This one is competitive. None of them are actually green, yeah. Okay, hopefully that helps a little bit. Okay. Yeah, on the actual computer screen, the colors are very different, but the projector is really good at making all colors look the same. Apologies. Okay. Any questions about that? All right. So, yeah. So now it is time to talk about our favorite virus. Yay! So, enzyme inhibitors have proved themselves to be really helpful for treating COVID-19 disease, which is caused by our favorite virus, SARS-CoV-2. And so one type of enzyme inhibitor that we can use that's used as an antiviral treatment are nucleotide analogs. So we've talked before about how SARS-CoV-2 is an RNA virus. So it needs a special RNA polymerase that helps copy its genome when it wants to replicate. And this is an RNA-dependent RNA polymerase, or RDRP. So there are drugs that you can use to inhibit the RDRP that basically resemble nucleotides. So the RDRP will start to incorporate these nucleotides as it's trying to copy the RNA genome. But you can design these nucleotide analogs to cause a chain termination. So it will just stop replicating and cause basically breaks or just an incomplete copy of the viral genome. So that means the virus can't make more copies of itself, and eventually it might die. So one type of nucleotide analog that has been used to treat SARS-CoV-2 is called remdesivir. Remdesivir, and I guess the other thing I should mention about nucleotide analogs is they're not specific to SARS-CoV-2, right? Any RNA virus that needs an RDRP, an RNA-dependent RNA polymerase, could potentially be affected by a nucleotide analog. And so remdesivir was actually first developed as an Ebola treatment, because Ebola is another type of RNA virus that uses RDRP. but remdesivir was already around when SARS-CoV-2 came along, and people tested it out and found that remdesivir is also effective against SARS-CoV-2. So what it basically does, I'm sorry, this picture is a little bit small, but on the top of this little diagram here, this is remdesivir. It's a nucleotide analog that looks a little bit like ATP. Well, the prodrug has this funny little thing here instead of 3 phosphates, but the rest of it kind of looks a little bit like adenosine. But it has, okay, so there's the interesting or useful thing about it is that it has a cyano group right here. And what the arrow group is pointing to the prodrug form. Your body will actually process the drug into the active form. That is the triphosphate form. So it looks just like a nucleotide triphosphate. And the main difference is that it has a cyano group right there that I just circled. And the cyano group, what it does is when it's incorporated into the growing RNA that's being copied, which is shown here, so this orange little, this looks orange to you, yellowy orange, is the RDRP. Do you have a question? It's, I would say it's a competitive inhibitor because it's competing for actual nucleotides at the active site of the RDRP. So, what will happen is the RDRP will take up remdesivir instead of actual nucleotides and incorporate them into the growing RNA chain. And actually 3 nucleotides later, so that's what's shown here. So remdesivir is R. It can be incorporated. It looks a little bit like A, so it matches up with the U, which is shown here. And then 3 nucleotides later, it causes a steric clash where nothing else will fit into the active site. It blocks it. And it does this sort of by distorting the structure of the RNA itself because of that big old cyano group. And ultimately, this will cause the RDRP to stall and eventually to stop copying. And something else that's interesting about it is that SARS-CoV-2 has basically a proofreading enzyme that will find mistakes as it's copying RNA and sort of remove nucleotides to try to fix the typos. And that's this purple enzyme shown here is the proofreading enzyme, the exonuclease. And so like this little scissor here is kind of showing it's trying to erase back to the remdesivir that got incorporated. But the remdesivir actually resists getting removed. So like the SARS-CoV-2 little proofreader can't fix the mistake once it's been incorporated. So that's pretty bad for the virus. Great for us, bad for the virus. This diagram also shows, I also wanted to say that remdesivir has been shown to be really effective against coronaviruses and also RSV, if you've heard of that one. They've tried it against flu. It does not work against flu. But since it's a general nucleotide analog, it can be potentially useful against any number of RNA viruses. And this was the first drug that was approved to treat COVID-19, mostly because it already existed when SARS-CoV-2 appeared, because it was developed a few years ago to try to treat a different disease. There's another nucleotide analog shown in this figure called bemnifosbuvir. This one is still in trials. It works very similarly to remdesivir. I won't go into the details of it. It looks more like GTP than ATP, and it works fairly similarly. So that, yeah. I guess not necessarily. I mean, because it's ribose based, right? So it's going to be incorporated into RNA. So I guess, yeah, it could be used for DNA viruses too. That's a good point because you need to make RNA out of them. But something to keep in mind is that you don't want a drug that treats a virus that will also hurt our own cells. So our own cells are also trying to copy DNA into RNA to make our own proteins. And we don't want to stop that from happening. We want to specifically stop the RDRP, which the virus needs, but we don't need. And so these types of nucleotide analog drugs are often chosen because they are more specifically going to be taken up by RDRP and not by like our own RNA polymerase. Because you want to inhibit the virus, but not inhibit ourselves. Because otherwise we'll have side effects, yeah. So that's remdesivir. There's another nucleotide analog that was also developed for COVID-19 called molnupiravir. This one looks more like CTP or UTP, and this one's a little bit different. So this one, instead of causing a chain termination where the RDRP gets blocked and it just stops, molnupiravir will actually encourage mutations. So once this little diagram sort of shows how that works. So once molnupiravir is taken up, it's shown here matching up with this over here is matching up with an A. So it should, once you make another copy of that, an A should match back up with the molnupiravir, but you can see here that a G got taken up instead. Over here it was matched up with a G, so it should match up with a G, and in this case it did. But basically what it does is it's confusing. It will base pair with not specifically with other nucleotides, that as you continue to make copies of the genome, mistakes will get incorporated. And over time, this is hopefully detrimental to the virus. You make enough mutations, you're going to have problems with your proteins functioning. And so this can be, Sorry? Yeah, essentially, yeah. And molnupiravir actually was also first developed for other types of viruses. It was first developed for hepatitis C. It's also been studied in flu and RSV and a number of other viruses. And it is approved for COVID-19. And so a lot of countries sort of made stockpiles of it. But eventually Paxlovid came out, which we'll talk about in a little bit. And Paxlovid sort of works better than molnupiravir, so people don't really use molnupiravir that much now. And now there's all these kind of stockpiles of it that are sitting there gathering dust, which we'll talk about in a little moment too. And so molnupiravir, it can be good or bad because it induces mutations, but they're unpredictable, right? So you could potentially make a mutation that actually makes the virus more fit or better at, spreading. You could make a virus that's less fit. So it's sort of a delicate balance, let's say, or it's less predictable than something like remdesivir that just straightforwardly stops replication. So one little note on molnupiravir, I just learned this the other day. I don't think this is in the slides I posted, but molnupiravir was recently found to be effective against another type of coronavirus. So in Cyprus, there are a lot of feral cats, and recently there has been a major outbreak of this coronavirus that causes feline infectious peritonitis, which is FIP. FIP is basically 100% fatal if it's not treated. And there's just been this massive outbreak. It's spreading to all these different feral cats and also to domestic cats. And so recently people have been studying if molnupiravir might be effective against this feline coronavirus that causes FIP. And it turns out it is really effective. So recently, Cyprus just approved molnupiravir for use in cats, which is fun. So now they can use up some of their stockpile of molnupiravir that's been sitting there because no humans really want it. And now the cats can have it, which I think is pretty great. So that's molnupiravir. And so now we're going to switch gears a little bit. So we were just talking about nucleotide analogs. These prevent the replication of the virus or cause mutations. Another type of really helpful enzyme inhibitor that we can use to treat COVID-19 are protease inhibitors. So I think we mentioned a while ago when we were talking about some of the different proteins that SARS-CoV-2 has. It has a protease. It's called Mpro or 3CL-Pro. And this is a cysteine protease. So we talked about serine proteases last chapter. This is a cysteine protease, so it works a little differently. But it's essential for processing of the proteins that the virus needs to, survive or to replicate and spread. And so if you're able to inhibit this type of protease specifically, it's really great because it stops the virus from packaging up little baby viruses and then sending them out into the world. And this is also another great target for a drug because we as people don't need this protease. So if we can specifically inhibit this protease, it should have very few side effects on, our proteases and our cells. And so So types of drugs that are also competitive inhibitors for these types of proteases can be really effective. And so one type of protease inhibitor that you may have heard of is called Paxlovid. Or Paxlovid, I should say, is a mix of two drugs. One of them is a protease inhibitor. So Paxlovid has been used pretty commonly to help reduce risk of hospitalization and death from COVID-19. Like I just mentioned, it's made from two drugs. One is called nirmatrelvir. That's the actual protease inhibitor. And so there's some links here about it. I also just threw in a picture of it. So this is nirmatrelvir by itself. And what it does is it forms a covalent bond with the cysteine in the enzyme active site and blocks the active site from binding to actual substrate. The other drug that's in Paxlovid is called ritonavir. Ritonavir is an inhibitor of cytochrome P450-3A4, which is in your liver and it helps to metabolize a lot of drugs, including nirmatrelvir. So ritonavir basically is there to slow your body down from breaking down nirmatrelvir so it lasts longer in your body. And I just threw in another little picture of nirmatrelvir in the active site of the Mpro enzyme. So it just fits right in there. It forms a covalent bond, which this arrow is pointing to. In part B of this figure, it's showing how it just fits really nicely in that active site and interacts with all those different amino acids that are in the active site. And here's that cysteine that it forms the covalent bond with. I also, I don't remember if I said, but the covalent bond that it forms is actually reversible. So it doesn't stay stuck in there forever. So that's why you kind of need to keep taking more Paxlovid until eventually you get rid of the virus. Yeah. It's the actual outline. So like the black lines that are showing the molecule, right? This is the drug. I won't outline the whole thing, but you got the idea. And then the little circles are all the amino acids in the active site. Any questions about those enzyme inhibitors and viruses? All right, so the next part of this questions we'll go over are about control of enzyme activity, but we are about out of time, so we'll finish up with that on Monday. The quiz for this will be due 2 Mondays from now, the Monday after we return from Thanksgiving break. Also, don't forget, the quiz for Chapter 11 is due this coming Monday, right before break starts. And was there anything else I wanted to tell you? Your exams are graded. Your scores are posted. I don't have them on me. They're in my office, so if anyone wants to take a look at their exam, they're in my office. I'll bring them on Monday, and then you can take a look at them then if you prefer. But that's it for today. So enjoy the rest of your day and your weekend, and I'll see you Monday.